Hi, I'm Sandy Kubillus. I'm an adjunct instructor in the biology department, here to talk to you about chemical water tests, the lab number 6. And let me share my screen and show you the PowerPoint that I have created with this. Okay, so you can see here is a very nice, clear running stream. Has a good ecology to it, clear water, probably high in dissolved oxygen, a fairly good stream. Unfortunately, many of our areas don't look like this. So we're going to talk about various chemical tests today. So we have oxygen in the water, which is called dissolved oxygen, and how does it get into the water? Well, we have various interactions with the air that's going to mix it in. So we have wave action, especially on very windy days. Lake Michigan obviously can have waves that are like up to 5 feet high, but little ponds and lakes are probably just going to have very small waves, although they do have whitecaps fairly often where they're mixing in with the air. We get photosynthesis from aquatic plants that are below the surface, and these plants will emit oxygen. And then many of our lakes and ponds, well, mostly ponds, have various fountains to them or bubbler systems to get more oxygen into the water because it helps improve the aquatic environment for fish, and also it's better, more aerobic environment, so it actually promotes the better ecology for the system, and that's why you see a lot of fountains, and especially the ones that just disturb a little bit of the surface. Those are little bubblers where you're actually getting oxygen down to the bottom of the pond. Another way that oxygen gets into the water is by actually, when the water temperature decreases, it holds more oxygen. It has to do with the partial pressure of oxygen and how much it is soluble in the water. So cold water can hold much more oxygen than warm water. And that can be a problem, especially in the middle of summer when we have very warm water and you can get low oxygen levels, and so sometimes fish will die in the middle of summer because of their stress, because the water is warm, and also they have just low oxygen levels. And then you can have thermal pollution, which sometimes can occur from power plants, although that's usually pretty well regulated, but there are times where they might release warmer water than what is allowed. They're only allowed to have discharge water that's a few degrees warmer than what the background water is. Another test that we will be running, and also very concerned with water quality, is nutrients. So mostly we'll be talking about nitrogen and phosphorus. You might remember these cycles that we had early in this semester. They're the key components of fertilizer. So I have a bag of fertilizer here, and you can see the 16 is the nitrogen level, and the 0 is phosphorus, and the last number 8 is potash, which doesn't really affect water quality so much. But nitrogen and phosphorus especially do, and actually it's very good to have 0 phosphorus in fertilizer. It took many years to get communities to try to enforce 0 phosphorus fertilizers because phosphorus is a major pollutant and causes algae blooms. And the algae then can cause other problems and sometimes cause a fish kill. So having low nutrients in our lawns and also in our farm fields is very critical. Now, lakes are actually categorized by how much nutrients they have. And there's numbers that go along with this, but mostly just the terms. And yes, you do need to know these terms. Oligotrophic is a lake with very low nutrients, like Lake Superior, which is very clear, it's cold, doesn't have a lot of aquatic plants in it. Mesotrophic is like Lake Michigan, moderate amount, meso, moderate, and Lake Michigan. Easy way to remember it, 3 M's. Eutrophic has high nutrient levels, and you might think of Lake Erie, so Erie, eutrophic. Most of our lakes in Lake County are this way, and in the later summer months, a lot of times you'll hear about Lake Erie having high algae blooms, which is affecting the water quality in some of the cities that take their water from Lake Erie. And then there's hypereutrophic, which is something caused by humans, and it's very high levels of nutrients. And that's kind of artificial because we use so much fertilizer, man-made fertilizer, and it's caused our lakes and ponds to become much more nutrient rich. They usually have abundant algae. Sometimes they have abundant aquatic plants that can prevent like even bringing a boat across because it's just the plants are just so dense. And eutrophication, so we just said eutrophic as Lake Erie. Well, eutrophication is the aging process that a lake goes through. It's a natural process. Normally it takes hundreds of years, thousands of years, depending on the size of the lake. Like the Great Lakes is taking many thousands of years, but humans have accelerated that dramatically. And eutrophication is how lakes will go from oligotrophic to eutrophic over time as they build up more nutrients. And humans have caused this to increase dramatically. Now, what I have here is a picture of blue-green algae. Just as a little warning, I like to put this as a little aside. That is, if you see a lake or a pond that looks like someone dumped bright green paint onto it, like this does, do not go swimming in it. And certainly don't let your dog drink from it. Some of these blue-green algae can have toxins associated with it, and dogs have been known to die from it. Humans have not died, at least in the United States, from it, but we usually don't drink water like this, and we usually don't go into it. But dogs, they will go into anything and drink anything. So beware. So we talked a little about why oxygen is important, and if you don't have enough of it, it's called hypoxia, where you have very low oxygen levels, and it can cause a fish kill. That's what the picture is about dead fish. And sometimes you can get a dead zone, which is an area where there's no oxygen, usually in a much bigger body of water than our local lakes, but like in the ocean. And we have a chapter of that in the textbook. I think it's 6.2, which talks about the dead zone at the mouth of the Mississippi River where it drains into the Gulf of Mexico. So high nutrients does not necessarily cause hypoxia. There's several stages, and students need to understand this a little bit better because high nutrients usually cause algae blooms. Not always, but most of the time will cause an algae bloom. And when the algae dies and decomposes, then the microbes that go to eat up that algae will then use up all the oxygen. And so that's what causes a fish kill. Also, if you have a very big algae bloom that's at the surface of the lake, it's going to block the sunlight from getting to those plants underneath it. So the aquatic plants that are growing under the water will die, and they're not producing oxygen anymore. And they're also contributing to the load for microbes to be eating up and using up more oxygen. So it's a major concern when you have these algae blooms. So we're going to be doing in this lab various chemical water tests. We have 9 of them. Some of them are very simple, some are much more complex. And you might remember from the pond lab, you looked at fish and macroinvertebrates, so the organisms that lived in the water. And now we're going to be looking at the water chemistry. So a couple of these things that we're going to be talking about, I'm going to go through the first 3 and then show you this various probe that I have that measures these. So pH is the acidity of the water, and it goes from 0 to 14 is the scale, and 7 is neutral. 7 is our drinking water. So anything below 7 is acidic, anything above 7 is basic or alkaline. And so 7 is neutral, and it's a logarithmic scale, so the further you get from 7, the much more acidic or basic it's going to get. And acidification is a concern because we have acid rain that raises the acidity of the water, and also from climate change because we have excess carbon dioxide in the water or in the air that's also going into the water when it rains, and it gets absorbed by the water. And the standard we have is from 6.5 to 9. So we'll test our samples in a minute on that. And then temperature is a concern mostly for very warm water. Some lakes and ponds I've seen get up to about 30 degrees, which is like in the mid 80s. It's very warm for water around here. Our samples, though, have been sitting out in the lab for a while, so they are basically at room temperature, so that's not really going to be much of an effect. And dissolved oxygen, we already talked about it. Dissolved oxygen has a standard of 5 milligrams per liter for fish, which also is based a little bit on different months of the year. That's partly because of the water temperature, but also partly about when fish will spawn, which is in the spring and early summer as the water just starts to warm up. So at this point, I wanted to show you my water samples. And here's my water samples. This is from the Des Plaines River, and this is from Willow Lake, which is what the pond lab was about. You can see they look fairly clear. They're not that turbid. You would expect the Des Plaines River one to be a little more turbid. And then I have a multi-probe meter. This is called a Quanta, and it measures dissolved oxygen, temperature, and pH. And a little bit faster than using the test kit. And I had a little bit of a mix up there. Okay, so you just would put this into the water sample and you could measure those parameters fairly quickly. In the lab, how we typically measure dissolved oxygen is with a test kit called a Hock test kit for dissolved oxygen, where you'd use various chemicals and water bottles. And it takes about 15 minutes. It's fairly long to do that. Okay, now we want to go to the next session of, I want to talk about hardness and some of the other components. So hardness, most people don't really know what it is. It's not ice. It's not hard water. It's not ice, although it technically is, it's frozen. But anyway, hardness is a measure of calcium and magnesium in the water. So it's these minerals that are in the water. And you get these from the groundwater that we have. When it goes through the rocks, it will pick up some of these minerals. And hard water is a concern mostly if you have well water and you get you'll get staining of your sinks and toilets. You'll get, you won't be able to lather too well with soap. So it's kind of an inconvenience. We can still drink it. There's a drinking water standard of 500 milligrams per liter. Our water is not that hard, but it can vary quite a bit from 0 to greater than 180 milligrams per liter. And but hard water is good in that it buffers the lake from changes in pH. Sometimes you get, if you get algae blooms, they can change the pH. It can raise the pH quite a bit, and this will buffer it somewhat. So I wanted to show you this one measurement because it's actually a little bit dramatic on the change for it. So let me stop sharing my screen. Okay, so this is for hardness, and we have our sample, which we collected using this 5 ml tube, and then we'll add this chemical to it, and you can see it changes it to a pink color. Then we're going to add this EDTA for hardness and count the drops. 3, 4, 5. I know it's going to take a fair amount to get it that high. 6, 7, 8, 9. Notice how I'm shaking it in between. 10, 11, 12. Oh, all of a sudden it's 12. Notice how it's no longer pink. So it's turned a bluish color. So that is our answer. It's 12, and I believe, based on this chart here, we need to multiply by 20. So that would be 240 is our hardness. Remember I said 180 is hard water. So this is 240, and that was the, I believe it was the Des Plaines River one. So you can see that is very hard water. Okay, now the next one is carbon dioxide. And we talked a little about carbon dioxide before, where we're exhaling carbon dioxide, and carbon dioxide is kind of influences the pH of the water. So it can make the water more acidic, and it should not exceed 10 milligrams per liter. And it's also due somewhat to climate change because of the excess CO2 in the air that is leading it to ends up getting into the water as well. And then the next one is turbidity, and that is the clarity of the water. You might remember in the pond lab, there was, they talked about Secchi disc a little bit. It's a round disc that's white and black, and that's used if you're out in a lake and you can drop it below the surface and see where it disappears and determine how turbid the water is. Well, this sample I have, this test kit's not quite so fancy as my multi-probe meter, and so it kind of is based on the Secchi disc. And basically a water is, a lot of times it'll be a brownish tint to it or a greenish tint. Greenish tint you really only get in the during the growing season in the summer because of algae, but if you have a brown tint, it's usually clay or silt that's been stirred up, and that could be from a rain event, or it could be because of fish, especially carp will dig into the soil and make it much more turbid. Water clarity is going to affect various plants. If it's too turbid, they can't get the sunlight again. It blocks the sunlight and will kill them off, and so they're not producing oxygen. And sometimes it can affect fish, especially sight-feeding fish. They may not be able to find their food. Sediment also can fall onto fish eggs and prevent them from hatching. So we only have a drinking water standard of 1 NTU. You have a glass of water, you can see right through it. It's very clear. There isn't a surface water standard. It has been very controversial because of construction sites, basically, that sometimes release water, turbid water into the environment. They're not supposed to have it get off their construction site, but sometimes that happens after extreme storm events where their silt fence and other things that they're using to control erosion just may not be working. So let me stop sharing my screen for a second. And this was the carbon dioxide sample, and it looks pretty clear. And you can see it has a very slight pink to it. So carbon dioxide is very minor change. It's difficult to see. Now, the turbidity has a dot in there, and that is like a little Secchi disc. So you'd fill this up with clear water, like tap water, and then you have your sample in another one, and you look down from above and add this solution that would make the clear water match the turbidity on here as you look at that black dot on the bottom. And it's kind of a basic, a little bit of a crude method, but it does work. It's not as fancy as a meter at this point, but at least it does work a bit. Okay, now the next couple that I want to show is chlorine, which is added to drinking water to kill bacteria. So, you know, you don't want to drink water that's right out of Lake Michigan, even that's where you probably get your water from, because you are afraid of the bacteria is going to make you sick and make you go, you know, have stomach upset. So there's a limit of 19 micrograms per liter or 0.019 milligrams per liter. And so it's mostly an influence because of what we add to the water to kill bacteria. So natural water bodies should have very little chlorine to it. Also, chlorine's a gas, so it does escape up into the air. We're going to also test for phosphorus and nitrogen. Phosphorus and nitrogen are nutrients, remember, from a few slides ago. And phosphorus also comes from animal droppings and from sewage. It can also come from the soil that might have had fertilizer in it. So the state standard for that is 0.05 milligrams per liter in lakes. And the nitrogen, we're going to look at ammonium nitrogen, which is from fertilizers again, and also from animal droppings, and that limit is 15 milligrams per liter. Now, we're not going to look at radium, which is radioactivity, which is a concern if you have groundwater, they're using groundwater wells, and sometimes you can have radium as a concern. And water softeners can help that. So let me show you a couple of things here. Okay, so with chlorine, that was a colorimetric test where you added some chlorine to these tubes, and then you had to look at this through a, look at the color changes, and there's like a little color wheel that's inside here, and you would look at the different changes that way. It's a very crude test. But then with phosphorus and also nitrogen, we have a little pocket colorimeter. And so here we had a base sample and a phosphorus sample that was from the Des Plaines River. Let me show you this. You can see this is quite blue, while this one is pretty clear. And so what I would do is put this in here and put the cover on it. Now we always want to standardize our samples first so that we know that everything we're seeing is because of the phosphorus and not because of any background coloring that there might be in the water. And then we would add the other sample, which is a test sample, and get the sample. Now this has been sitting around for a while, so it might be a little bit different, but it's reading 0.53, which actually I believe is about what we had for Willow Lake. So there's a little pocket colorimeter. That's something similar for ammonia nitrogen. And notice we had two different colors here. We have a greenish color. Let me get that where you can see it over the white. And that was the higher sample. That was the Des Plaines River, and this was Willow Lake. And again, you would put these samples in here, close it up, and read your sample as well. So that was just a quick way to show you how these tests run, because if you were doing these in the lab, it would take you the entire lab period. It takes quite a bit of time to go through all these. So let me show you what we got as results. Okay, so here are results, and I have our standards in here as well, so you can compare them. Notice Des Plaines River and Willow Lake, they're both at room temperature. Dissolved oxygen, they were both about the same, and they were greater than 5 milligrams per liter, so we were fine. Carbon dioxide was a little bit higher in the Des Plaines River. Why this was 20 versus 10, I don't know. I normally don't test carbon dioxide when I run water quality testing. pH was fine. It was less than 9. And hardness was less than 200 milligrams per liter, but remember it's much higher than the 180, so 200 and 240, so they were both very hard samples. And turbidity was quite high, 35 versus 20. Des Plaines River was much more turbid. And our drinking water standard is 1. Remember, there's not a surface water standard. And phosphorus, this is the key one. Notice that phosphorus is very high. It's at 90.9 milligrams per liter, which is almost 20 times higher than the state standard. And the lake was also very high, about 7 times the state standard, so very high. And ammonia nitrogen, these both were under 15, so these are very good. And chlorine was 0, which is kind of what we expect for samples that are natural and not something that's, been treated already from a sewage treatment plant or something. So anyway, this was the water quality lab, and hopefully you'll understand what I was all talking about, and maybe in the future you'll be able to run some of these tests yourself.